So if you want to calculate the phenotypic ratio of dihybrid cross, you just have to multiply with the same number. So now phenotypic ratio of dihybrid cross. This is very important because even if they give you some questions will be out of NCRT, few very difficult questions will be from out of NCRT. So here this type of questions can be asked. So here you should not waste the time, just you have to multiply. If they give you any ratio, just multiply. So 3:1 multiplied by 3:1. How much will get? 9:3:3:1. So this is the phenotypic ratio of dihybrid cross. So just mono, you'll get the 3:1. So this is the monohybrid phenotypic ratio. Just you have to multiply again 3:1. 3 3s are 9. 3 1s are 3. First here, I'll put the arrow mark. 3 3s are 9. Again, 3 1s are 3. Again, take this number. 1 3s are 3. 1 1s are 1. So you'll get 9:3:3:1. Similarly, if we go for genotype, can anyone tell me what is the genotypic ratio of monohybrid cross?
1:2:1.
It is 1:2:1. Similarly, just you have to multiply 1:2:1 multiplied by 1:2:1. So if you multiply, you'll get the ratio 12, 24, 12, 12, 1. So you can remember like this, or just you can multiply. So this is a very easy number code to remember the genotypic ratio: 12, 24, 12, 12, 1. Fine. So this is the genotypic ratio of monohybrid cross, and this is the genotypic ratio of dihybrid cross. So you should not waste time during examination calculating the genotypic ratio, because it will consume more time by counting the progenies of the F2 generation. So you have to just directly put these numbers. And in problematic part, if they give you, if they give some different ratio, just multiply that ratio. You will get the phenotypic ratio of dihybrid and genotypic ratio of the dihybrid cross. Just you have to multiply. And then you need to remember about the allele. So one more thing. So we had discussed till dihybrid, right, and the ratios. So next is a very important topic: incomplete dominance. So do you have any doubt till this mono dihybrid cross in genetic parts terminologies we had discussed, right? Do you have any doubt in that?
No, ma'am.
You have understood completely. Now moving on to this topic: incomplete dominance. So basically, incomplete dominance, here what happens? So the dominant gene. So what did I tell you about the dominant gene and the recessive gene? Dominant gene will not allow the recessive gene to express itself. For example, if along with the dominant gene capital T, if small t is present, capital T is for tallness. So small t is for dwarfness. So if it is having these two genes, which character will be expressed? The F1 hybrid is tall or dwarf?
Tall.
If both the genes are there. Why the plant is tall, why not dwarf? Because this gene is also present, right? Small t. Why it is not dwarf? Why only tall? Tell me.
Because capital T
Only
is
the plant
dominant
is
over
tall. Why
small
not
t.
dwarf? Yes.
Because
Who is
capital T,
answering?
capital T is dominant over the small t.
Very good. Capital T is the dominant gene and small t is the recessive gene. As this capital T is masking the, what do you call it, as the small t. It is not allowing the small t to express itself. That's why this dominant gene capital T is expressed and small t will be suppressed. But in case of incomplete dominance, So the dominant gene, it is failing to what do you call it as the control the recessive gene. As a result, intermediate character is been obtained. So this is first found in which plant? So they will ask you, incomplete dominance was first found in which plant? It was in 4 o'clock plant. They will ask you the name of the plant. 4 o'clock plant. So, what is the scientific name of the 4 o'clock plant? Mirabilis jalapa. Mirabilis jalapa. This is 4 o'clock plant. So generally, गुलाबश का झाड़ करके बोलती नहीं. Have you all heard of this plant? Gulabash. If they get swelling and all, they'll bring the leaves of that plant and just heat and put it over that. Um, so all that pus will come out from that wound. So with yellow flowers, with red flowers. So that is Mirabilis jalapa. So now, apart from this, so later on, it was also found in some other plants. So that is in snapdragon. In your textbook, they have given the example of the snapdragon, which is also called as dog flower, antihirum. So, but first incomplete dominance was found in Mirabilis jalapa. So, in NEET, if they ask you, the first incomplete dominance was found in which plant? So, you should not write snapdragon. It was in Mirabilis jalapa. Later on, after experimentation, they found out even in snapdragon also, you'll find this incomplete dominance. So, now here, here the parent, you'll have red flower and male. If you take red flower, the female, let it be a white flower. So these are the P1 parents. So here the plant with the red flower and plant with the white flower. So the what are the possible gametes? Small r, small t. So these are the gametes of P1. And then in F1 generation, you'll get capital R and capital T. According to the law of Mendel, what should be the phenotype of this F1 hybrid?
Red.
Tell me. So we have studied the laws of Mendel. According to laws of Mendel, that is the law of segregation, law of what do you call it as the law of dominance. Which color should be the F1 hybrid? What should be the color
Red.
of F1 hybrid?
Red.
Sorry,
Pink.
Red.
I'm telling you. I'm just comparing it with the law of Mendel.
Pink color.
According to Mendel, the dominant gene should express itself, right? So don't
Red
forget
color.
about incomplete dominance. In case
Red.
this plant is obeying law of very good. Who answered me?
Me.
Who gave me this answer? Red.
Me.
Your name, good name, please.
Umra. Umra Adil.
Umra, very good. So, if it obeys the law of Mendel, it should be red. But this plant is disobeying the law of Mendel, so the F1 hybrid will be pink in color. Because red is blending with white and you'll get an intermediate character that is pink. Then we'll go for selfing of F1. If we go for selfing of F1, that is capital R. So you can take either small r or small t. Okay, so I have taken small t, you can take small r. Okay, so in your, why did I take here t? Because in your examination they can change this alphabets and they can give you, but you have to follow the same thing. Okay, so here small r, here capital R, small r. Okay, letters can be changed and can be given. So now if we go for selfing of F1, so capital R, small r, cross of capital R, small r. So what are the possible gametes here? Capital R, small r, capital R, small r. We'll put it in the checkerboard or the Punnett square. Capital R, small r, capital R, small r. Capital R, capital R, capital R, small r, capital R, small r, small r, small r. So what is the phenotypic ratio?
1:2:1.
Very good. Who answered this?
Mummy.
Your good name, dear?
Ikhra.
Ikra. Very good, Ikra. 1:2:1. How many red?
100.
Red.
How many pink?
Two pink.
Two pink
One white.
and one white.
White.
So if you have capital R, then it will be red. If you have capital R, small r, then it will be pink. Blending takes place, and then if it is both small r, then it will be white. One red, two pink, and one white. And what will be the genotypic ratio?
3:1.
No,
1:2:1.
no.
9
How
is
come
to 3 is to 3 is to 1.
No, dear, this is monohybrid. So don't get confused between a monohybrid and a dihybrid. This is a monohybrid cross. Because we are just taking one character, red and white. Only one pair of contrasting character we are taking.
3:1, ma'am.
3 is to 1.
1:2
Okay.
is to 1.
You are telling 3 is to 1. How? How come genotypic ratio is 3 is to 1? Tell me, what is 3? How much? What is 3? What is 1? You should tell me now.
3 red
Ma'am,
and 1 white.
1:2 is to 1.
So, first of all, tell me the difference between what is phenotype and genotype. You're getting confused, my dear. What is the difference between phenotype and genotype? Can anyone answer me? What is phenotype and what is genotype?
Phenotype is all the possible
Ma'am,
external visible
1:2
characters.
is to 1.
Very good. So now,
Genotype
huh,
is the genetic makeup of an organ.
so
Substance
genetic
organ.
makeup means what you have to take? You have to take the genes, genetic constitution. So now tell me how come it is 3 is to 1? How come the genotypic ratio is 3 is to 1? How much is 3? How much is 1 in this?
3 red and 1 white.
You cannot tell colors, my dear, in genotype. You have to tell about the genes. Capital R, capital R, capital R, small r.
1:1, ma'am. 1:1.
Okay, you told me 1 is to 1. How come you are
Ma'am,
telling 1 is to
1:2
1?
is to 1.
Sorry? 1 is to?
Ma'am, 1:2 is to 1.
Very
2 is
good.
to 1.
Very good. 1 is to 2 is to 1. Who gave this answer? 1 is to 2 is to 1?
Ahad.
1 is to 2 is to 1. It's the right answer. Because you are having one capital R, two capital R, small r, and one small r. Yes? So this is wrong. This is also wrong. This is right. In case of incomplete dominance, phenotypic ratio is equal to genotypic ratio, which is equal to 1 is to 2 is to 1. If they ask you about the phenotype and genotype ratio of the incomplete dominance, just close your eyes, write the ratio 1 is to 2 is to 1. Phenotypic ratio is equal to genotypic ratio. In need, they can ask you like this. In which type of inheritance the phenotypic ratio will always be equal to the genotypic ratio? That is in case of where?
Complete dominance.
Incomplete dominance. Did you understand why did I put phenotypic ratio 1 is to 2 is to 1? Why? Because you had one red
1
flower,
red, 2 pink,
two
1 white.
pink and one white. Good. And why is the genotypic ratio 1 is to 2 is to 1? It is 1 capital R, 2
One
capital R, small r, and one small r, small r.
Smaller.
That's the reason the genotypic ratio is 1 is to 2 is to 1. Did you understand?
Yes, ma'am.
So this is the concept of incomplete dominance. So they will ask you the name of the plant where the incomplete dominance was first discovered. So they can ask you what do you call it as the um, uh, some other plant apart from Mirabilis jalapa if they ask you. So your answer should be the snapdragon.
Snapdragon.
Huh. And who observed this phenomenon, incomplete dominance? You should remember the name of the person, Karl Korens. Make a note of this, Karl Korens. Once they have asked Karl Korens. So I said you about the name of the three persons who rediscovered the work of Mendel. Who rediscovered? Karl Korens, Teskmark, and Hugo de Vries. Okay. So now here, with respect to this incomplete dominance, who observed this incomplete dominance? Karl Korens. Karl Korens. C double C square. You can remember. So he was working on snapdragon or the dog flower. In that he had observed Karl Korens. Fine. Did you understand? So anyone
Yes,
have
ma'am.
doubt in this the concept of incomplete dominance?
No, ma'am.
Okay. Next, the most frequently asked question is about test cross. So what is this test cross? It is a cross between F1 hybrid and recessive parent. Cross between F1 hybrid and the recessive parent is what you call it as the test cross. Supposingly you have a tall plant. So you have crossed tall plant, dwarf plant. So gametes capital T, small t. Here F1 hybrid is capital T,
Capital T, small
small.
t, small t.
Now if you are making a cross, so which one is the F1 hybrid? Tell me over here. Which is the F1 hybrid? Which is the F1 hybrid here? Which is the F1 hybrid? What is the full form of F1 hybrid? F1 means what? They
First
will ask you.
filial generation.
First
First
filial
Parental
filial.
generation.
generation.
Now, what is the F1 hybrid? So this have labeled here. It is there itself. What is this F1?
Capital T, small t.
Capital T, small
Very
t.
good. Capital T, small t. Cross between F1 hybrid and the recessive parent. You are making a cross. So this one is the F1 hybrid. And which is the recessive parent?
Small t, small t.
Very good. Small t, small t. So this is the dominant parent. This is the recessive parent. So small t, small t. So what are the possible gametes from here? Capital T, small
Small
t.
t.
Now here only one possibility.
Small
Now
t.
we'll put this in the checkerboard. Capital T, small t. Now here you just have small t. Capital T, small t, small t,
Small t,
small t.
small t.
So the ratio is 1:1. One capital T small t and one small t small t. So this is the monohybrid test cross ratio. Likewise, you can uh, um, do the test cross for the dihybrid cross also similarly. Right? Did you understand? So this 1:1 is what? Monohybrid test
Cross
cross
ratio.
ratio. Monohybrid
Phenotypic
test cross
ratio.
ratio. Did you understand my point?
Yes.
So,
Yes, ma'am.
yes, um, once they had asked, so out of NCRT related to this cross itself, so test cross is mentioned in your syllabus, but they had asked about back cross. Okay, so now back cross is test cross. You have understood, right? You're crossing the F1 hybrid with the recessive parent is the test cross. So in case of back cross, it is a cross of F1 hybrid with any of the parent. If you are crossing the F1 hybrid with either dominant parent or recessive parent, it's not like no rules, no regulations. You can make a cross between either this or this, either dominant or recessive parent, then it is called back cross. Did you get my point? F1 hybrid with either dominant parent or either recessive parent, it is called as
Back
what?
cross.
Backwards.
Back
Backwards.
cross. Crossing of F1 hybrid with either of the parents. It could be with capital T or small t. So you can tell test cross is a type of back cross. Yes?
Yes.
It is a type of back cross because you're crossing. Back cross means you can cross with either dominant or recessive. In test cross, you're crossing the F1 hybrid with the recessive parent. So it is test cross is a
Back
type of back cross. Okay, so most probably they will ask you about test cross only. But in case of assertion and reason, if they ask you test cross is a type of back cross, your answer should be true. So that's why I'm making you clear. Did you get my point? So this mind this what do you call it? This point should be clear in your mind that it is a type of back cross. Test cross is a type of back cross. Clear? Okay. Now, what is the importance of test cross? This question was repeated. So, why are you going to do test cross? There will be some reason, no? Because of that, you are performing this test cross. It is basically performed to find out the genotype of this F1 hybrid. To find out the genotype of F1 hybrid. So the test cross is being performed. So in case of the options, they will give you. So test cross is conducted. Reason. In options, they will give you 4 types. In that, the correct answer is to find out the genotype of F1 hybrid. Understood? So this is the
Yes.
main reason. So you have to remember this. Okay? Is it clear?
Yes, ma'am.
Okay. So then apart from this, we have the multiple allelism. You can ask me doubts if you have any doubts in this test cross because it is a very important
No, ma'am.
topic. You have any doubt?
No, ma'am.
Ma'am,
Yes, ma.
uh, is there any phenotypic or genotypic ratio in test cross?
Phenotypic ratio, see. So here you will get tall. Here you will get dwarf. If you go for phenotypic ratio, what is the phenotypic ratio? Tell me. How many tall?
One
One.
tall, one dwarf.
One.
One is
One
one.
is to one.
How many dwarf? One.
One.
So the phenotypic ratio is 1:1. If we go for genotypic ratio, 1 capital T is to 1 small t, which is also equal to 1:1. Even here, the phenotypic ratio and genotypic ratio is same, 1:1. Clear?
Yes.
Huh. Next. Next, anything else? You have any doubt in this topic?
Ma'am,
Yes, ma.
does all the process which is
Hmm.
regarding to monohybrid is equal of both phenotypic and genotypic ratios are equal?
In which thing? In which thing? You are asking about the incomplete dominance or the test cross or the monohybrid cross. In which thing the
All
genotype
the
and phenotype?
process of all the process of monohybrid has the equal phenotypic and genotypic
You
ratios.
know, in case of monohybrid cross, the phenotypic ratio is 3:1. Genotypic ratio is 1:2:1. Right? Because there you will get all the 3 tall plants and only 1 dwarf plant, isn't it? Only in the monohybrid cross, here the phenotype and genotype will be different. But if you go for incomplete dominance and the test cross, the ratio will be same. The phenotypic and genotypic ratio will be same. In the normal pure what do you call it as the monohybrid cross, the plants which are obeying the laws of Mendel, the phenotype and genotype will be different. If they don't obey the law of Mendel, if there is deviation from Mendelism, then the ratio will be same. Understood?
Okay,
So
ma'am.
now if we talk about the normal monohybrid cross, the ratio will be different. Phenotypic and genotypic ratio will be different. If we go for incomplete dominance, the phenotypic and genotypic ratio will be same. If we go to test cross, the phenotypic and genotypic ratio will be same. Clear?
Okay, ma'am.
Next. And they will ask you whether this snapdragon is a pure breeding stock. Karke they can use this terminology. Whether this snapdragon or dog flower is a pure breeding stock. This once they had asked. Is the dog flower or the snapdragon which shows incomplete dominance, is it is a pure breeding stock? Your answer should be no. Why it is not a pure breeding stock? Because like if we go for if we take its F1 generation, capital R small r, it is in heterozygous condition, isn't it? And there is mixing and blending of 2 characters, red and white. So it cannot be a pure breeding stock. Okay? So you have to remember this term, whether it is a pure breeding stock or no. When I go to what do you call this, the first law, when I had explained tall plant and a dwarf plant, we took 2 characters, right? Tall and the dwarf. In that, the F1 hybrid, so whether it is an pure breeding stock or what? The plants which obey the law of Mendel, they are considered to be pure breeding stock. The plants which do not obey the law of Mendel, they are not a pure breeding stock because they will be mixing and blending of characters. Clear? So you remember this term, pure breeding stock. The antiherum, it's not a pure breeding stock. The F1 hybrid, it's not a pure breeding stock. Done? You have understood? Shall we move on to the next topic?
Yes.
Then we'll discuss about the multiple allelism. Again, one more topic, interesting topic, and a very important topic for NEET, multiple allelism. What is this multiple allelism? So the expression of genes in more than 2 allelic forms is what do you call it as the multiple allelism. So see, in the name itself you can understand multiple allelism. One gene, it is having many allelic forms. Gene is having what? Multiple allelic forms, then it is called as the multiple allelism. So it can be best explained by using ABO blood grouping. So the blood grouping in human beings, it is called as the ABO blood grouping. So by you taking this ABO blood grouping, you can clearly understand the multiple allelism. So here the blood group, it is being determined basically by the antigen which is present on the RBC. Suppose this is your RBC. What is RBC?
Red blood cells.
Huh. On your RBC. So this is your RBC. Imagine this is your RBC. That is your red blood corpuscle. On this RBC, there is a antigen. There is a antigen present on your RBC, and the type of the antigen present on your RBC, it will determine the blood group. So now this synthesis of this antigen which is present on your RBC is being regulated by a gene. And what is the name of the gene? I. Are you understanding my point? The antigen which is present on your RBC is determining the type of the blood group, and the synthesis of this antigen which is present on your RBC is being determined by what? It is determined by the gene I. And what is this I representing? It is representing isoagglutinogen. This is very important. Make a note of this. I represent isoagglutinogen. So now this gene I, it should have many alleles. That is the concept of multiple allelism, right? One gene should exist in multiple allelic forms. Now this gene I, it expresses in how many allelic forms? 3 allelic forms. So that's your understanding over here. What is the meaning of multiple allelism? One gene expressed in terms of many allelic forms. Now this gene I is expressed in how many allelic forms? 3 allelic forms. Now what are the 3 allelic forms? IA, then
IB. IB.
IB,
IO.
then just I.
IO.
Okay, you can write IO or just I, one and the same. So this IA, it is coding for antigen A. And this is coding for antigen B. And this doesn't code for either antigen A or antigen B. A or B. So now how many alleles are there for gene I? 3 allelic forms are there. And then that is IA, IB and IO. So now how many phenotypes are there in the blood group and how many genotypes are there? So phenotypes are 4. Here you should remember this number. They will ask you. Genotypes are 6. So how come phenotypes are 4? How many types of blood groups do you have?
AB. ABO.
Phenotype, I will represent it by P. Okay. Phenotype A, B,
AB.
and
O.
O. So these are the 4 phenotypes. 1, 2, 3, 4. And what are the genotypes? For blood group A.
IA.
How
IO.
many genotypes?
IA. IO.
IA,
IO.
IO.
So these are the 2 possible genotypes. For blood group B,
IB,
IB,
IB.
IB, then
IB.
IB.
Two possible genotypes. Now, blood group AB, how many possible genotypes are there?
IA, IO, IB.
IB. Any other possibility for blood group AB?
No.
No, ma'am.
Sorry? Any other possibility?
No, ma'am.
No possibility. Why? Because it is codominant. You require both the genes. If A is absent, you'll have blood group A. B is absent, you'll get. If sorry, B is absent, you'll have blood group A. A is absent, you'll get blood group B. So if you want blood group AB, you will require both the genes. Then for blood group O,
IO, IO.
so now how many possible genotypes? 1, 2, 3, 4, 5, 6.
6.
Fine. So phenotypes are 4, genotypes are 6. You have to remember this number, very important. So they will ask you in case of multiple allelism, phenotypic versus genotypic ratio 4:6. Phenotypic ratio is 4, genotypic ratio is 6. And how many alleles are there for the gene I? How many alleles are there for gene I? 3 alleles are there. Don't get confused with the phenotype and genotype with the allele. If they ask you allele, your answer should be 3. If they ask you phenotype, your answer should be 4. If they ask you genotype, your answer should be 6. Did you get my point?
Yes.
So now, this is about the concept of this multiple allelism. Then there is one more concept which you call it as the codominance. Codominance. What is this codominance? So here, in case of dominance, one gene is dominant and another is recessive. In incomplete dominance, the dominant gene fails to mask the recessive gene. Whereas in case of codominance, so both the genes are dominant. For example, just now I taught you about the blood group AB. Yes? Here, can you tell any one of these genes are recessive or dominant in blood for blood group AB?
Dominant.
Both are dominant. So without B, A is useless. It cannot produce blood group AB. Without A, B is useless. It cannot produce the blood group AB. To produce blood group AB, it should have both the genes IA and AB. So that's why it is considered as codominant. Did you understand? So it is a phenomenon where two dominant alleles they express independently. Okay. So for example, the blood group AB, the genotype IA and IB should express to produce the blood group AB. So this is an example of codominance. Like even in case of animals, if you take, so you could have seen, like if you cross a cow with the ox is black in color, cow is completely white in color, but the offspring will have patches of both black and white. It's not the mixture of black and white producing gray. It is having black patches along with the white patches. Black patches from the father and white patches, white color patches, it's because of from the mother. If it is not a codominance, then you could have got, you will get a elephant color, no, gray color, black and white mixing together to produce gray color. But if the offspring is getting both black and white color, then it is called as the codominance. Did you understand what is codominance?
Yes.
So this is also, so in while they can ask you like, by just giving an example, why both the characters are being expressed in this offspring? Why not only one character from the parent? So your answer should be, this is because of the concept of codominance. So that codominance will be there in the option. You have to mark that. So they can give you any example where two characters are expressed. Both the characters of the parents are expressed equally. Okay? Okay, so this is about the concept of the codominance. Next important concept is pleiotropy. So now to understand this pleiotropy, can you tell me what is multiple allelism? Just now I had explained you about multiple allelism.
They have
One gene,
multiple.
Many alleles controlling
To form three allelic forms.
three allelic forms will be there. So for just one character, that is blood group, you are having how many alleles? You are having many
three
alleles.
alleles. Three
So
alleles.
opposite to multiple allelism is the pleiotropy. So here gene is one. It is just a one single gene, and that is producing many phenotype. Phenotype is many. One gene, one allele, it is responsible for the production of many characters. So there many alleles are producing one character is multiple allelism. Did you get my point? So you are having, in case of blood groups, how many alleles? How many alleles are there? Many alleles are there, right? In blood group, how many alleles did I tell you?
One.
One allele, three alleles.
Three
Yes,
alleles.
for multiple allelism, three alleles. Right? For blood group, three alleles. Three alleles are producing one character. That is your ABO blood group. ABO blood group. But if one allele, it is producing many characters, then it is called as pleiotropy. Opposite to multiple allelism is pleiotropy. Pleiotropy, many alleles, one character. In pleiotropy, one allele, many character. You have to remember like this. Did you get my point? Multiple allelism, many alleles, one character. In pleiotropy, one allele, many characters. Did you get my point?
Yes, ma'am.
Huh. So the best example here, they will ask you example PKU, example for phenyl keto, example for pleiotropy, PKU and starch synthesis. Remember this example, starch synthesis in pea seeds, starch synthesis in pea seeds. So now I'll explain you by taking this phenylketonuria. So if the person is having phenylketonuria, then he will get many characters. What are the different characters? Means symptoms. Many symptoms he will get, like mental retardation, that is one character or symptom. Then reduction in hair, that is the second character or the symptom. And skin pigmentation, okay, and mousy yellowish colored urine, fourth character. So you are getting so many character. That is because of mutation of one gene. Mutation of one gene, that is coding for the enzyme PAH. It is mutation of one gene, that is resulting in the mutation of one gene resulting in the decreased synthesis of PAH enzyme, and it is resulting in how many symptoms? Around 4 to 5 symptoms. So that is what you call it as the pleiotropy. So this disease is also very important. You need to understand what is this phenylketonuria. So full form of PKU is phenylketonuria. Here one enzyme will not be synthesized. So what does this phenylalanine hydroxylase enzyme it will do? So this phenylalanine hydroxylase enzyme, it will convert phenylalanine to tyrosine. Phenylalanine, it gets converted into tyrosine in presence of which enzyme? PAH enzyme. But the people who are suffering from this phenylketonuria, there will be no PAH. If there is no PAH, this phenylalanine will remain as phenylalanine, and then slowly this phenylalanine will get converted into phenyl pyruvic acid, and this phenyl pyruvic acid, it is toxic to central nervous system. So that's why they become mentally retarded. There will be reduction in hair, they will have skin pigmentation and so many problems. And why this PAH is not synthesized in this phenylketonuria people? Because one gene which is responsible for the synthesis of this PAH enzyme, it is defective. It is mutated. That's why one gene is producing many symptoms. What are the many symptoms? Mental retardation, reduction in hair, skin pigmentation, and so on. Around 5 characters, 5 symptoms are being produced. And that 5 symptoms, it's because of how many genes? How many genes are producing the 5? One gene, which is responsible for the synthesis of which enzyme? Phenylalanine hydroxylase enzyme. That is defective. That's the reason how many characters are produced? 5 characters, five symptoms are being produced. So this is what you call it as the pleiotropy. Did you understand my point? One gene producing many character. Pleiotropy, many genes producing one character, multiple allelism. Okay, here you have to remember PKU, full form of PKU, phenylketonuria. And the people who are suffering from phenylketonuria, they are called as PKU idiots. And what is the full form of PAH? Phenylalanine hydroxylase. Clear? You have any doubt?
No, ma'am.
Okay. So then, this pedigree analysis is also very important. So there you have to remember the symbols of the pedigree analysis. So what is this pedigree analysis? The analysis of traits or the diseases in different generations of a family is what you call it as the pedigree analysis. Like if you are analyzing a trait or if you are analyzing any disease in different generation. So तुम्हारे में एक डिसीज है, तुम्हारे बच्चों में नेक्स्ट जनरेशन जाते रहे चाहिए, सब में एनालाइज करे तो एक 3 से 4 जनरेशन को तुम्हें एनालाइज करे तो उसे हमें क्या बुलाते हैं? पेडिग्री एनालिसिस. जैसा कि क्वीन विक्टोरिया, उनके फैमिली में क्या था तो हीमोफीलिया करके एक था डिसऑर्डर, जेनेटिक डिसऑर्डर. उनके पूरे फैमिली मेंबर्स में वो पूरा क्या करको ही स्टडी करको ही, उसे हमें क्या बुलाती हैं तो पेडिग्री एनालिसिस. अब यहाँ पे जो है पेडिग्री एनालिसिस में थोड़े सिंबल्स जो ही वो सिंबल्स नीट में पूछती, वो सिंबल्स को तुम्हें अच्छे से याद रख लेना. ओके, अब जैसा कि ये पूछ के थे नीट में ऐसा 2 लाइन्स डाल के ऐसा. सो दिस सिंबल वाज बीन गिवन एंड व्हाट डज दिस इंडिकेट करके पूछे थे. ओके, सो दिस आई विल टेल you. First you should understand the basic symbols, then this can be understood. So this was the neat question in which they had given the symbol and you have to answer to this symbol. What is the meaning of this symbol करके you have to answer. So now तुमना basic symbols क्या है, what are the different symbols मैं तुमना पूरा बता डाल चु, तब जो है तुम्हें अच्छे से इसे आंसर कर सकती. अब नाउ दिस सर्कल आया तो फीमेल, बॉक्स आ गया तो स्क्वायर बॉक्स आ गया तो देन मेल। अब तुम ना समझ में आ गया दिस इज फीमेल दिस इज मेल करके। एंड इफ आई पुट लाइक दिस then this means it is an unknown sex. You don't know whether it is male or a female, and if you don't know, then you'll put like this. Then if I put the box similarly like this, and if I shade completely, then it is an affected male. If I circle and if I shade, then it is affected female. अब ये जो सिंबल में है मैं शेड करके हूं क्या?
No, ma'am.
यहाँ पे शेड करके हूं?
No, ma'am.
हाँ, तो इसका मतलब मेल एंड फीमेल ही दोनों भी अनअफेक्टेड ही नॉर्मल ही। ठीक है, अब ऐसा मैं एक सर्कल ड्रॉ करी, इनके बीच में एक ही एक लाइन डाली। ये सिंबल का मीनिंग क्या कर तो मेटिंग, मेटिंग और मैरिज। अब तुम ना एग्जाम में पूछ के ही सेम देखो ही डायग्राम बट 2 लाइन्स डाल के ही। व्हाट इज द मीनिंग ऑफ दिस सिंबल? कॉन्सैजिनस मेटिंग, कॉन्सैजिनस मेटिंग, CONSANGUINEOUS मेटिंग। सो नाउ व्हाट इज द मीनिंग ऑफ दिस कॉन्सैजिनस मेटिंग? इसका मतलब मेटिंग बिटवीन क्लोज रिलेटिव्स। अब रिश्ते में शादी कर डालती नहीं, मामा की बेटी उनकी फूफू की बेटी, वैसा रिलेशन में कर डालती नहीं, क्लोज रिलेशन में शादी कर डालती नहीं, राइट? वैसा अगर रिलेशन में शादी होके है देन दैट इज कॉन्सैजिनस मेटिंग। व्हाट इज दिस कॉन्सैजिनस मेटिंग? मेटिंग बिटवीन द क्लोज रिलेटिव्स और द मैरिज बिटवीन द क्लोज रिलेटिव्स। अगर एक लाइन आया तो जस्ट मेटिंग, 2 लाइन आ गया तो कॉन्सैजिनस मेटिंग। ये आके पूछ के थे, ये सिंबल दे डाल के व्हाट इज द मीनिंग ऑफ दिस करके। आया समझ में?
Yes, ma'am.
ओके, नेक्स्ट अगर एक सर्कल डाले उसमें खाली एक ही एक डॉट डाल के ही, इसका मीनिंग क्या है का तो कैरियर फीमेल। यूजुअली मेल्स कैरियर नहीं रहते, फीमेल्स इज कैरियर रहते। सो एक डॉट डाल के रहते हो, क्या बोलना उसका मीनिंग क्या है? कैरियर फीमेल। एंड नेक्स्ट क्या है? सो इफ आई पुट लाइक दिस, अब देखो दो भी सेम डायग्राम्स इच ओके फर्स्ट अब इसका मतलब क्या है? इसका मतलब क्या है? देखो डिफरेंस थोड़ा अच्छा है, इसमें यहाँ पे लाइन डाल को हूं, इस पे यहाँ पे नहीं डाल को हूं। ठीक है, इनो का तो डाइजोगोटिक ट्विंस इनो, यू नो मोनोजाइगोटिक ट्विंस। एक लाइन यहाँ पे बीच में डाल डाली को तो मोनोजाइगोटिक ट्विंस। सो डू यू नो द डिफरेंस बिटवीन मोनोजाइगोटिक ट्विंस एंड डाइजाइगोटिक ट्विंस? अगर किसी को मालूम है तो बोलो। मोनोजाइगोटिक ट्विंस का और डाइजाइगोटिक ट्विंस के बीच में क्या डिफरेंस कर को बोलो। व्हाट इज द डिफरेंस?
Here there is one zygote, here 2 zygote.
हाँ, इसमें भी क्या होता है तो जाइगोट स्प्लिट होता, बट यहाँ पे क्या होता है तो मोनोजाइगोटिक में एक स्पर्म एक एग फ्यूज हो जाता, फ्यूज हो को क्या बनता? जाइगोट बनता। वो जाइगोटा को स्प्लिट हो जा को 2 जाइगोट बन जाती। आया समझ में? डाइजाइगोटिक में क्या होता है तो 2 स्पर्म्स 2 एग्स, यू नो 2 जने अलग स्पर्म अलग एग फ्यूज होके 2 अलग अलग जाइगोट बनती। यहाँ पे जाइगोट स्प्लिट हो रहा, यहाँ पे जाइगोट स्प्लिट नहीं हो रहा। सो नॉर्मली 1 एक एग प्रोड्यूस करती, सो बट इन फ्यू केसेस क्या होता है 2 एग्स क्या होती है, एवॉल्व होके मेच्योर हो जाती हैं, 2x से साइमल्टेनियसली मेच्योर हो जाती हैं. You know that many sperms enter, right? It's not just one sperm is entering at a time, millions of sperms they are entering at a time. So those two sperms they come in contact with the 2x and two babies are formed. डाइजेगोटिक ट्विंस में चांसेस रहते हैं मेल एक फीमेल एक, बट मोनोजाइगोटिक ट्विंस में रहे तो दो भी मेल्स रहते हैं या फिर दो भी फीमेल्स ही रहते हैं. कई को बोलो व्हाट इज द रीजन?
Split.
बिकॉज इट इज जस्ट वन जाइगोट. एक स्पर्म में जो एक्स क्रोमोसोम है वाई क्रोमोसोम है ऑलरेडी डिसाइड हो गया रहता, वो जाइगोट स्प्लिट हो रहा. सो वो वजह से इधर बोथ ऑफ देम विल बी मेल और बोथ ऑफ देम विल बी फीमेल. अच्छा मैं यहाँ पे और एक ड्रॉ कर चू, एक पेडिग्री का सिंबल, तुम्हें बोलना वो क्या करके. ओके यू हैव टू टेल मी. इट इज रिलेटेड टू ट्विंस ओनली. समझ में आगा नहीं मोनोजाइगोटिक और डाइजाइगोटिक ट्विंस में डिफरेंस क्या है करके? यू हैव अंडरस्टूड राइट? समझ
Yes,
में आया
ma'am.
नहीं आया?
Ma'am.
हाँ, अब आई विल ड्रॉ वन मोर दिस पेडिग्री symbol of related to twins. You have to tell me whether it is a monozygotic twin or a dizygotic twin. So tell me.
Monozygotic.
मोनोजाइगोटिक.
Sorry,
मोनोजाइगोटिक.
कैसा होगा मोनोजाइगोटिक? मैं यहाँ लाइन ड्रॉ करको हूँ बीच में.
बोथ आर
Dizygotic.
फीमेल्स.
Ma'am,
Both are females, but did I draw the line in between?
नहीं मैम.
हाँ, लाइन ड्रॉ नहीं करे तो इट इज ए डाइजेगोटिक ट्विन. डाइजेगोटिक ट्विन में एक गर्ल बेबी एक बॉय बेबी हो सकता है, नहीं तो दो भी गर्ल बेबीज भी हो सकती. अच्छा देखो यहाँ पे मैं ऐसा ड्रॉ करी हूँ. अब इसके बीच में जो है लाइन डाली में. अब ये क्या हो गया?
Monozygotic.
मोनोजाइगोटिक.
Monozygotic twins both are females, dizygotic twins both are males. Okay, now I'll draw one more. What is this?
Dizygotic.
डाइजाइगोटिक.
Dizygotic twins both are
Male.
मेल.
male. Okay, now one more symbol I will draw here. Same.
Monozygotic.
Monozygotic twins
Male.
Male.
male. Both of them are males. Monozygotic twins में दो पॉसिबिलिटीज हिंस हो. क्या बोलो? दोनों लड़के रहती दोनों लड़कियां रहती. कई का तो एक ही स्पर्म है ना, स्पर्म में एक्स क्रोमोसोम रहा तो फीमेल्स हो जाती हैं, स्पर्म में वाई क्रोमोसोम रहा तो लड़के हो जाती हैं. वो जाइगोटिक स्प्लिट हो रहा. सो ओनली टू पॉसिबिलिटी इन मोनोजाइगोटिक ट्विंस. डाइजाइगोटिक ट्विंस में एक लड़का एक लड़की हो सकती, या फिर दोनों लड़कियां हो सकती, या फिर दोनों लड़के हो सकती. ये तीन पॉसिबिलिटीज हैं किसमें? डाइजाइगोटिक ट्विंस में. एक स्पर्म में एक्स क्रोमोसोम था और एक स्पर्म में वाई क्रोमोसोम था, तो एक लड़का एक लड़की हो गए. दोनों स्पर्म में वाई क्रोमोसोम थे तो दोनों लड़के हो गए. दो स्पर्म में भी एक्स क्रोमोसोम था, दोनों लड़कियां हो गए. तो तीन पॉसिबिलिटी ही डाइजाइगोटिक ट्विंस में. मोनोजाइगोटिक ट्विंस में ओनली टू पॉसिबिलिटीज. अंडरस्टूड? In the next class, we will work out few problems on pedigree analysis. तुम ना पेडिग्री चार्ट देती, वो पेडिग्री चार्ट को एनालाइज करको ऑटोसोमल डोमिनेंट ट्रेट है या फिर ऑटोसोमल रेसेसिव ट्रेट है करके तुम्हें एग्जाम में लिखना पड़ता. So that I will teach you in the next class. So have you all understood the concept whatever I have taught you today? All these are very important topics. समझ में आया? सिंबल्स पूरा समझ में आए
Yes, ma'am.
पेडिग्री एनालिसिस के? अब नेक्स्ट क्लास में हमें चार्ट स्टडी करेंगे.
Ma'am,
Okay.
when was next class?
So that will be updated in the group.